In Module 5, all of the viruses that we're talking about are single-stranded, positive-sense RNA genome viruses. But the first two families that we discussed, picornaviridae and cheliciviridae, were all naked virions. With the togaviridae and the flaviviridae, these viruses, although still positive-sense, single-stranded RNA, these viruses have an envelope, so that makes them different. And they're interesting families with a lot of important pathogens in them. They used to be different genera in one family, but they've since been separated into their own families. Let's talk about the togaviridae first. The family name comes from the Latin word for cloak, or toga, because of the envelope. It's a family of small envelope viruses with single-stranded positive-sense RNA genomes. Within the family, the alpha virus genus includes a large number of species that are mostly mosquito-borne and pathogenic in their vertebrate hosts. Many are important human and veterinary pathogens. Alpha viruses get their name from the Greek letter alpha because they were once group A arboviruses. Members of this group grow easily in cell culture, and there are many animal models, so this group has been really well studied. Now we have the rubevirus also on this slide. This is the virus that causes German measles. It infects only humans, and transmission is person to person. Before April 2019, the genus rubevirus was in the family Togaviridae, but it's since been moved into its own family. We won't worry about that for our purposes, though, because your book is still using the old classification. Now let's look at virion structure. Remember, these viruses have a lipid envelope surrounding the nucleocapsid, and inserted in the viral envelope are virally encoded glycoproteins. We've talked about the envelope glycoproteins before and their roles as anti-receptors and so on. Now, if you look at the structure of togaviridae, you can see that there are these large glycoprotein spikes. There's 80 of them in total, and if you look at the illustration here, you'll see that the capsid proteins are in direct contact with the envelope proteins. Now, the envelope proteins are trimers. That means that they're made up of 3 parts, and each part forming the trimer is a heterodimer. A dimer is 2, and a heterodimer just means that the 2 parts making up the dimer are different. So E1 and E2 proteins form these heterodimers. So those are the 2 proteins that form the dimers, and then 3 of the dimers will come together to form the trimeric glycoprotein spike. The nucleocapsid is icosahedral in symmetry with T equals 4. When we look at the genome organization, remember it's a positive sense, single-stranded RNA genome. It's arranged in 2 modules. The 5' proximal 2/3 codes for non-structural proteins that are required for replication and transcription of RNA. Remember that because the virus has to make RNA from RNA, it's going to need to code for its own viral enzymes to do that, because the eukaryotic cell is not going to have those kinds of enzymes for it to use. The genome has a 5' cap and a non-coding region that's followed by the non-structural and structural genes. Then, so you can see two open reading frames. The first, remember, contains the non-structural genes, and the second open reading frame codes for the structural genes. At the end of the RNA, at the 3' end, is a poly-A tail. The negative sense RNA of the replication intermediate is neither capped nor contains a poly-A tail, so we only see that in the genomic RNA. There is a subgenomic RNA that's used to translate the structural proteins. That's transcribed from the negative sense RNA, and it also has a 5' cap in poly-A tail. So we'll talk more about the subgenomic RNA in a bit, but this is the basic structure of the genome. And so we have the two open reading frames with the replicase genes, the non-structural genes, loaded at the proximal end. The receptors for alpha viruses are most likely ubiquitous cell surface molecules that allow the viruses to attach to both insect and vertebrate cells. So keep in mind, the viruses in this family have a very wide host range, and most of them cycle between insects that are the vectors and then vertebrate animals that are the hosts. The virions are brought in through endocytosis, and fusion between the viral envelope and the endosomal membrane is triggered in the low pH environment. So this is similar to what we had talked about before. For penetration and uncoating, again, the attached virus particles are taken up by receptor-mediated endocytosis or viropexis. The virus particles fuse with the endosomal membrane, and then the nucleocapsid is uncoated to release the RNA genome. When we look at the proteins that are on the viral envelope, remember there's E1 and E2 that form the heterodimers. The viral glycoprotein E2 mediates receptor binding. They've done experiments where they see that antibodies directed against E2 will neutralize the infectivity of the virus. Also, when they've found amino acid mutations in E2, those mutant viruses had an altered efficiency of binding and virulence. So these observations suggest that E2 is the anti-receptor. We see that a drop in pH inside the endosomal vesicle causes conformational changes in E1 and E2, and in particular, a fusion domain in E2 is exposed. And this allows for fusion of the viral envelope and the vesicle membrane, and that leads to the release of the nucleocapsid into the cytoplasm. In terms of transcription and translation, translation of genome RNA begins at an initiator UAG near its 5' end. It usually terminates at a UGA codon positioned close to the end of the non-structural P3 coding region, generating a P123 polyprotein. Complete P1234 protein forms only when there is a read-through of this codon, and that only happens about 10 to 20% of the time. Then that protein will undergo a series of cleavages to yield the individual non-structural proteins. So when we look at this, again you see that the 5' proximal ORF, or open reading frame, is going to be responsible for translating the non-structural genes. And keep in mind that the replication strategy for a single-stranded positive sense RNA virus is going to be that the first step is going to be translation. Because it's a positive sense RNA, it can be directly translated into proteins. And so that's what we see happening here. The first step is going to be translation of those non-structural proteins, and those will give rise to the protease and the polymerase and so on, so that you can then shift over to transcription. Eventually, there's going to be a shift from translation of non-structural proteins to transcription of RNA. The non-structural proteins carry out synthesis of the different viral RNAs, a highly regulated event in infected cells. During the early phase, full-length negative-strand antigenome RNAs are synthesized. At this phase, little or no positive-strand RNA is produced. In contrast, during the late phase, mostly positive-strand genome and subgenomic RNAs are made. The switch from synthesis of negative to positive strands is correlated with proteolytic processing of P123. Cleavage is not autoproteolytic, so it only happens when the concentration of P123 becomes high enough so that the molecules encounter each other and undergo mutual transcleavage. So if you look at this slide, you can see that I've got some numbers indicated on it, 49S and 26S. The S refers to Svedberg units, which is a sedimentation coefficient. It's just an indication of size of the different species of RNA. So the genomic RNA, the input positive sense RNA, is 49S. A full-length minus strand RNA transcript is made of that. But then you'll find that there are two species of positive sense RNAs that are transcribed from the negative sense RNA. One is 49S, a full-length copy, and the other is 26S, a shorter copy, and this one is used for translating the structural proteins. So let's look at this again, just to maybe make it a little bit more clear. I made this slide. So the input RNA is the positive sense 49S RNA. That's what enters the cell when the virion is entered, uncoded, and the genome enters the cytoplasm of the infected cell. The first step, because remember, this always happens with positive sense single-stranded RNA viruses, you're going to get translation of non-structural proteins. Included in these non-structural proteins will be the polymerase, which can then switch the process over to transcription, because ultimately you have to make more RNA if you're going to make progeny virus. So you get a shift when you have a sufficient concentration of non-structural proteins. You get the shift to transcription of the negative sense RNA. Remember, you have to go through that negative strand as part of the replication scheme because it's complementary. So you make a full-length negative sense RNA. Now this negative sense RNA is transcribed, but it can be transcribed into two different species of RNA. It can be transcribed into a full-length positive sense RNA, a 49S RNA, and that's the RNA that can be encapsulated into progeny virus particles. But there's also a 26S RNA, and that is used strictly for translation of structural proteins. So the capsid, E1 and E2. So the antigenome RNA is the template for the synthesis of both full-length and subgenomic positive strand RNAs. At the peak of virus formation, about 10 times more subgenomic RNA than genome RNA has accumulated. Making higher levels of subgenomic RNA is one means by which the virus is able to produce more structural than non-structural proteins. And that's important because many of the non-structural proteins, such as the polymerase, are enzymes which can be reused, whereas the structural proteins, remember the capsid and the envelope proteins, you have to have many, many copies of each of those proteins to make up each individual virion. In addition to being produced in large amounts, the subgenomic messenger RNA, which encodes viral structural proteins, is also very efficiently translated. During translation of subgenomic messenger RNA, the capsid protein is released from the growing polypeptide chain by self-cleavage. Capsid proteins associate and bind to genome RNA in the cytoplasm to form nucleocapsids. The remainder of the structural polyprotein is translocated as a unit to the endoplasmic reticulum by means of a signal sequence at its N-terminus. Proteolytic processing produces PE2, 6K, and E1. Additional protein modifications are carried out during vesicle transport to the cell surface. But this is the process, because remember, capsid proteins are not glycosylated, so they're going to be directly translated in the cytoplasm. But the envelope proteins, those are glycoproteins. They have to move through the ER and Golgi, and that's not only necessary for their glycosylation, but it's how they get inserted into the membrane. So this is important to remember, that the capsid proteins are strictly in the cytoplasm, the envelope proteins go through the Golgi and the ER. In vertebrate cells, capsid proteins and genomes assemble in the cytoplasm. Capsids associate with E proteins at the plasma membrane where budding occurs. In the case of animal cells, infection is usually cytopathic. However, in insect cells, the infection can be non-cytopathic. In this case, the virions probably bud into intracytoplasmic vesicles and are released by exocytosis. Large amounts of virus are made in animal cells, resulting in the high titer viremia that facilitates ingestion of the virus by blood-feeding insects. So having a high level of virus circulating in the blood ensures that when the hematophagus insect feeds on the host, they will acquire the virus and become infected. This is an overview of togavirus replication in vertebrate cells. So these are all the steps that we've talked about. You have attachment of the virion to host cell receptors, the particles brought in by endocytosis. You have a drop in pH, which leads to fusion and uncoating. You get translation and RNA replication. The glycoproteins are going to be translated in rough ER and get processed and carried to the plasma membrane, where they're going to become inserted. They're going to fuse with the plasma membrane of the host cell. Capsid proteins are assembled in the cytoplasm. The genomic RNA is inserted into the capsid, and they migrate to the plasma membrane and bud through. The alpha viruses replicate in both vertebrate and insect cells. In vertebrate cell lines, alpha viruses replicate very rapidly, with the release of progeny virus typically within 4 to 6 hours after infection. Infection causes extensive cytopathic effects in the vertebrate cells. In contrast, replication in insect cells is non-cytopathic, producing a persistent infection. So as I mentioned, alpha virus replication is relatively fast, resulting in acute disease in vertebrate hosts and strong cytopathic effects in vertebrate cell culture. And again, contrast this to what we see in mosquito cells, which results in persistent infection and shows little to no sign of fitness loss in infected mosquitoes. This indicates that there's a need to shut down general translation as a means to inactivate the vertebrate immune system, but this is not needed in insect cells. It has become clear that alpha viruses not only cause a general host shutoff in infected vertebrate cells, but also specifically suppresses different host antiviral pathways using their viral non-structural proteins NSP2 and NSP3. So this is, when we look at this makes sense from an evolutionary standpoint, because these viruses are using two very different types of hosts. And with the vertebrate, the key is that the vertebrate host develops a high level of viremia, circulating viremia in its blood. So we see cytopathic effect and very rapid replication in the vertebrate host. In the mosquito, it's essential that the mosquito remain infectious for life. A mosquito will only feed a couple of times in its life history, and so it's essential that if it becomes infected during one blood meal, that it remains infectious long enough to transmit that virus during its next blood meal. So we see persistent infections, and it's also critical that the behavior of the mosquito not be affected. It has to be able to continue to go out and find hosts and so on. So we see that alpha virus infection of the invertebrate does not cause a change in fitness of the insect. Alpha viruses are transmitted to humans through the bite of mosquitoes in a cycle involving vertebrate reservoir hosts. The viruses are subgrouped according to the prevalence of the clinical symptoms they cause in humans. The encephalitic alpha viruses occur in the Americas and are associated with severe and lethal encephalitis. This group includes the Venezuelan, Eastern, and Western equine encephalitis viruses. The arthritogenic group causes incapacitating and long-lasting articular disease and myalgia and comprises the Ross River virus, chikungunya virus, Sinbis virus, Myaro virus, Onyongyong virus, and Barmaphorus viruses. These viruses are globally distributed and are responsible for endemic diseases in some regions. The virus is delivered extravascularly by an infected mosquito and infects local muscle cells or Langerhans cells in the skin. Langerhans cells can carry virus to local lymph nodes. The virus is delivered to the blood and spreads to target tissues such as skin, joints, and the central nervous system, in addition to distant muscle and lymphatic tissue. Now let's consider a few diseases that fall into this group. First, let's consider Eastern equine encephalitis virus, or triple E. The incubation period for triple E, which is the time from the infected mosquito bite to the onset of illness, ranges from 4 to 10 days. Triple E virus infection can result in a systemic febrile illness or neurological disease, including meningitis, which is the infection of the membranes that surround the brain and spinal cord, or encephalitis, which is actual infection of the brain. The type of illness will depend on the age of the person and other host factors. Some people who become infected with triple E may be asymptomatic, that is, they don't develop any symptoms at all. Systemic infection is characterized by fever, chills, malaise, arthralgia, and myalgia. The illness lasts 1 to 2 weeks, and most people recover completely when there's no central nervous system involvement. Signs and symptoms of neurological disease include fever, headache, vomiting, diarrhea, seizures, behavioral changes, drowsiness, and coma. In infants, neurological disease often occurs soon after onset. In older children and adults, encephalitis may occur after several days of systemic illness. Approximately a third of all people with encephalitis due to triple E infection die. Death usually occurs 2 to 10 days after onset of symptoms, but can occur much later. Of those who recover, many are left with physical or mental sequelae or after effects, which can range from mild brain dysfunction to severe intellectual impairment, personality disorders, seizures, paralysis, and cranial nerve dysfunction. Many patients with severe sequelae require long-term care and die within a few years. The enzootic and epizootic epidemic transmission cycles of triple E are shown in this slide. The enzootic triple E virus transmission cycle is maintained between passerine birds as a reservoir, they're the amplification hosts, and the mosquito Culicida melanura. That's the main enzootic vector, and this mosquito lives in swamp habitats. Various rodents and marsupials may serve as principal enzootic vectors and reservoirs in South America. Passerine birds develop extremely high levels of viremia, enough to infect both enzootic vectors as well as a variety of bridge vectors. Humans and equids are dead-end hosts since they do not develop sufficient viremia to transmit the virus. About 30% of the 103 triple E cases recorded in the last decade have occurred in 2019. On average, there are about 11 human cases of triple E in the United States each year. Triple E virus has infected humans in 22 U.S. states, mostly around the Gulf Coast, in the Great Lakes region, and along the Atlantic coast. Conditions that encourage mosquitoes to proliferate can increase the disease risk, such as warmer temperatures. So one of the reasons we're seeing outbreak cycles more frequently is probably related to the fact that the mosquito breeding season is longer now. That's cause for concern given that climate change is pushing average temperatures up across the globe. From 2010 to 2019, most of the cases of triple E have been reported from Massachusetts, Michigan, Florida, Georgia, New York, and North Carolina. Triple E transmission is most common in and around freshwater hardwood swamps in the Atlantic and Gulf Coast states and the Great Lakes region. Chikungunya virus is spread to people by the bite of an infected mosquito. The most common symptoms of infection are fever and joint pain. Other symptoms may include headache, muscle pain, joint swelling, or rash. Outbreaks have occurred in countries in Africa, Asia, Europe, and the Indian and Pacific Oceans. In late 2013, chikungunya virus was found for the first time in the Americas on islands in the Caribbean. There is a risk that the virus will be imported to new areas by infected travelers. There is no vaccine to prevent or medicine to treat chikungunya virus infection. Travelers can protect themselves by preventing mosquito bites. When traveling to countries with chikungunya virus, use insect repellents, wear long sleeves and pants, and stay in places with air conditioning or that use window and door screens. Now let's talk about rubella virus, or German measles. This is a member of the genus Rubavirus. In this case, man is the only host, so we don't have an arthropod vector. Transmission is from droplets shed from the respiratory secretions of infected people. People may shed virus from 7 days before the onset of the rash to approximately 7 days after rash onset. Transmission from mother to fetus can also occur, with the highest risk of congenital rubella syndrome, or CRS, if infection occurs in the first trimester. Infants with CRS can transmit virus for up to a year after birth. Rubella is usually a mild disease with rash, enlargement of lymph nodes, slight fever, and a long incubation period. The incubation period can last as long as 3 weeks. Complications are rare and more common in adults. However, there's a high incidence of congenital deformities in infants born to mothers infected during the first trimester. These deformities include deafness, cataracts, and microcephaly. Persistence of virus in the infants with CRS has been reported. They've been able to isolate virus from these persistently infected infants as long as 3 years after birth. It's thought that this could be a survival mechanism for virus in nature. Endemic rubella virus transmission was declared eliminated in the Americas in 2015. However, rubella virus continues to circulate widely, especially in Africa, the Middle East, and South and Southeast Asia. There is a very effective vaccine to control rubella. It's the R in the trivalent MMR vaccine, or measles, mumps, rubella. Now, before April 2019, this genus was included as part of the Togaviridae, but now it's been moved to its own family, the Matonaviridae. We won't worry about that for this course because obviously that's a very new circumstance, and I want to stay consistent with what's in your textbook. Rubella is a very dangerous disease for a pregnant woman and for her developing baby. Anyone who is not vaccinated against rubella is at risk of getting the disease. And although rubella was declared eliminated from the U.S. in 2004, cases can occur when unvaccinated people are exposed to infected people, mostly through international travel. During the decade occurring between 2005 and 2015, 8 babies with CRS were reported in the United States. So it still can and does occur here in the U.S. Women should make sure that they are protected from rubella before they get pregnant. Pregnant women who contract rubella are at risk for miscarriage or stillbirth, or their developing babies are at risk for severe birth defects with devastating lifelong consequences. CRS can affect almost everything in the developing baby's body. And in the slide up at the top left, you see what the rash looks like in a normal, uncomplicated case of rubella. So in this case, the child became infected after birth. But you can see the cataracts that can develop and the skin lesions that can develop in children that have CRS in the other 2 pictures. Now let's move on to the family Flaviviridae. This is a large family of positive sense single-stranded RNA viruses. It's enveloped. Until 1986, Flaviviridae was a genus in the family Togaviridae, but they've since split it out. And there are 3 genera now in this family: the flavivirus, the passcivirus, and the pestivirus. And don't worry, we'll talk about all of these. The family members have very diverse biological properties, but they do share similarities, of course, in the morphology of the virion, the genome, and so on. There are at least 73 viruses that are in this group that affect humans and are transmitted by arthropods. So again, we have a situation where many of the viruses in this group have an arthropod vector associated with them. In terms of physical characteristics, the virus has a small nucleocapsid with T=3 symmetry. It's enclosed within an envelope. The E protein is a transmembrane protein that binds to the cell surface receptors and directs fusion of the viral envelope with the host cell membranes. The M protein is a smaller transmembrane protein. So you can see the 2 different size proteins here in this illustration. And on the slide here, I said surrounded by a lipid bilayer, readily inactivated by organic solvents or detergents. That's a common characteristic of any virus that has an envelope. The organic solvents, detergents, will disrupt that envelope. So very early in virology, that was one of the first tests that would be done to try to determine whether a virus might be naked or enveloped. So this isn't characteristic just of the flaviviruses, but any virus that has an envelope. There are 3 viral structural proteins. We have the E protein, the envelope protein. That's the anti-receptor. It binds to cell surface receptors, and as I mentioned, it directs fusion. The matrix protein is smaller, but it's also a transmembrane protein, so that's associated with the virus envelope as well. And then we have the capsid protein. So those are the 3 structural proteins that we find with these viruses. Flaviviruses have 7 non-structural and 3 structural proteins. The structural proteins are designated E, PRM/M, and C for envelope, precursor of the membrane or membrane, and capsid proteins respectively. The precursor M protein associates with the E protein in the endoplasmic reticulum to form a heterodimer. This association apparently protects the E protein from a premature conformational change that would lead to exposure of the fusion peptide in the low pH environment in the Golgi. At a late stage in maturation, a protease cleaves PRM, causing a conformational change in the virion. A single capsid protein will assemble to form the capsid, and we have the 2 glycoproteins that are associated with the envelope. And what we see is that the envelope proteins, the glycoproteins, lay flat on the surface of the mature virion, giving it a smooth golf ball-like appearance. This structure of the envelope glycoproteins is pH sensitive. Under low pH conditions, they'll form spikes, and the spikes will mediate a membrane fusion. Members of the family Flaviviridae have positive sense single-stranded RNA genome. Members of the genus Flavivirus have a methyl G cap protecting the 5' end of their genomes. However, pestiviruses and hepacivirus genomes are not capped. The 3' end of the genomes of all of the members of the family have a hairpin structure that stabilizes the genome and is probably required for genome replication. Members of this family do not have polyadenylated 3' ends like we saw with other viruses. Between the 5' and 3' non-coding regions, or NCRs, of all members of the family is a single long open reading frame encoding a polyprotein. A single capsid and 2 or 3 envelope proteins are encoded at the 5' end of the open reading frame, while the RNA-dependent RNA polymerase is encoded at the 3' end of the ORF. Despite intensive research, the hunt for specific flavivirus receptors has yielded diverse results. A plethora of molecules at the plasma membranes of different cells have been shown to interact with flaviviruses that were proposed to function as attachment factors, but bona fide entry receptors are still ill-defined. The reported data are quite varied, suggesting that molecules involved in flavivirus cell attachment and entry differ among viruses and cells. In most instances, the major envelope protein E, which mediates viral membrane fusion upon receptor-mediated endocytosis, has been implicated in such interactions. More recently, cellular lipid receptors and other kinds of receptor families that recognize lipids in the viral membrane have been shown to mediate flavivirus attachment and entry in certain instances. In addition, we find that there are a number of flaviviruses that can enter through a process called antibody-dependent enhancement. And with antibody-dependent viral enhancement, what we see is a situation where a non-neutralizing antibody can bind with the virus, opsonize it or decorate it. That virus with the attached non-neutralizing antibody can bind with the Fc receptor on a monocyte, and that will allow the monocyte to bring the virus particle in, but in this case, rather than killing the virus is actually able to infect and replicate within the cell. So this is a situation where if you have an antibody of poor quality, perhaps directed against a different serotype, so that it doesn't completely neutralize the virus, that could actually enhance infection by providing another mechanism of entry into the cell. And this is of interest, particularly with dengue virus, which we'll talk about in a bit. So we see subneutralizing titers of antibodies that cause enhancement, and that allows for enhanced uptake of cells expressing the immunoglobulin Fc receptors. And this could in turn lead to enhanced disease. For instance, with dengue fever, when someone is infected a second time with a different serotype of dengue, so that their existing antibodies are not good neutralizing antibodies, we actually see a more serious form of the disease called dengue hemorrhagic fever. E proteins are important for cell infection as they mediate binding to cell receptors and fusion with endosomal membranes after host cell endocytosis. To help fusion with the endosomal membrane, E proteins undergo yet another structural change triggered by pH. In low pH, the E protein dimer dissociates and becomes a trimer with an exposed fusion peptide at one tip. Due to the essential role E proteins play in entering the host cell, they are the main target of anti-flavivirus antibodies, which attempt to block entry into the cell by hindering attachment in endocytosis or membrane fusion. So here again, the process of absorption, we have the binding of the virus to the appropriate host cell. It's brought in by endocytosis and clathrin-coated pits. So receptor-mediated endocytosis, which we've talked about many times. Within the endosome, you have the protein pump bringing hydrogen ions in, which results in a lowered pH. Lower pH causes the E protein to rearrange, and we see an exposure of the fusogenic domain. And that allows the virus envelope to bind to the envelope of the endosome, and that allows for the nucleocapsid to be released into the cytoplasm. Once the nucleocapsid is released, then we have uncoating, and the RNA genome is released into the cytoplasm for translation and replication. After virus uptake and release of the genome RNA into the cytosol, the RNA is bound by ribosomes and translated, giving rise to the polyprotein. Proteolytic processing yields 10 mature virus proteins. The 3 structural proteins are first. The viral polyprotein is inserted into the membrane of the endoplasmic reticulum. Then the capsid is cleaved off and released into the cytoplasm. The process of translation initiation varies by genus. The hepaciviruses and pestiviruses have an internal ribosome entry site, or iris, in the 5' NCR, while the members of the genus Flavivirus use cap-directed translation. The protein expression strategy of the members of the family Flaviviridae is to generate the majority of their proteins from a single polyprotein precursor by a series of stepwise proteolytic cleavages. This is very similar to the protein expression strategy that we discussed for the picornaviruses. Some products of the polyprotein remain cytosolic. For instance, the capsid protein and several of the non-structural proteins, while others, for instance, the envelope proteins, cross the ER membrane during synthesis. Cytosolic regions of the precursor polyprotein are cleaved by virally encoded proteases, while cleavages in the ER are accomplished with host cell proteases. Viral proteins residing in the ER lumen traffic through the ER and the Golgi, where they're glycosylated. The non-structural proteins, NS1 through NS5, are involved in the replication of the genome RNA. The synthesis of non-structural proteins results in the establishment of active RNA replicase complexes. RNA replication requires the synthesis of a minus strand copy of the plus strand RNA. This is followed by the synthesis of plus strands that are either translated, making more virus proteins, replicated, making more RNA, or packaged, making virions. So the same process that we've talked about with all of the single-stranded positive sense RNA viruses. The synthesis of RNA is asymmetric, generating more than 10 times as many plus strands as minus strands. The replication of the viral RNA takes place inside virus-induced invaginations known as vesicle packets, or VPs. Flaviviruses assemble their virions inside the cell by using intracellular membranes for envelope formation. Assembly of the nucleocapsid takes place at the cytoplasmic face of membranes with which PRM and E proteins are associated. Virus buds into the lumen of the ER and forms disorderly arrays of membrane-bound vesicles, as you can see in the EMs on this slide. Molecular details leading to flavivirus viral RNA encapsulation and assembly into new virions remains largely unknown. Notably, the process is thought to be highly specific, as only a single full-length positive sense viral RNA molecule is packaged into virions, whereas minus sense RNAs and host RNAs are excluded. This is a much more efficient, or I should say accurate, process than what we see for encapsulation for many other viruses. Nucleocapsids have not been visualized in flavivirus-infected cells, suggesting that encapsulation is rapid and or concurrent with replication and budding. The coupling of protein synthesis, RNA synthesis, and virion assembly on membranous structures assures that newly synthesized genome RNA can associate with capsid protein and initiate the assembly process. Encapsulation of the RNA initiates the budding of particles into the ER. Particles that have budded into the ER are then processed as they proceed through the Golgi. PRM is important in this process. Virions follow the exocytosis pathway and are released to the extracellular space. Cleavage of the PRM protein just prior to virion release converts the particle to mature form. So this slide takes us through the entire process of replication for the flaviviridae. So let's go through these steps. The virus binds to the surfaces of susceptible cells through interaction with attachment factors, and this binding is followed by interactions with secondary receptors that mediate internalization by endocytosis. Alternatively, in the presence of non-neutralizing antibodies, opsonized viral particles can be internalized through Fc receptor-mediated endocytosis. Endosome acidification triggers E-trimerization and fusion of the virus in endosome membranes, thereby releasing the nucleocapsid into the cytosol. The viral genome is then translated and processed before replication commences. Nucleocapsids are formed and assembly is initiated by budding into the endoplasmic reticulum lumen. The immature viruses follow the secretory pathway in which the decreasing pH of the trans-Golgi network triggers maturation through furin protease-mediated cleavage of PRM. Nascent virions are then secreted from the cell, and PR is released from the viral surface. Now let's go through the three genera within the family Flaviviridae. First, let's talk about the genus Flavivirus. There are about 80 members in this genus. They're almost all arthropod-borne. Many of them are significant human and or veterinary pathogens. And some of them have major global importance, including dengue virus, yellow fever virus, Japanese encephalitis virus, and West Nile virus. There are vaccines available for some of these, including yellow fever and Japanese encephalitis virus and tick-borne encephalitis. but we don't have vaccines for dengue or West Nile and many of the other members of this group. Also, there's no good treatment for these viruses. Patients are getting supportive care, making sure that they have fluids and so on. But we don't have specific antivirals against these. Ribavirin has been used, but it's been just so-so in its effectiveness. So it's really important for us to understand their replication, because that's how new treatments and new antivirals are going to be developed. Flaviviruses can be phylogenetically distinguished by their mode of transmission, that is, mosquito-borne, tick-borne, and those with no known vector. The mosquito-borne flaviviruses revealed two distinct epidemiological groups. The neurotropic viruses, often associated with encephalitic disease in humans or livestock, tend to be vectored by Culex species and have bird reservoirs. Then we have the non-neurotropic viruses associated with hemorrhagic disease in humans, and those are correlated with Aedes species as the vector and primates as the hosts. Thus, the tree topology describing the virus-host association may reflect differences in the feeding behavior between Aedes and Culex mosquitoes. The tick-borne viruses also form two distinct groups, one group associated with seabirds and the other, the tick-borne encephalitis complex viruses, associated primarily with rodents. The correlation between epidemiology, disease correlation, and biogeography begins to define the complex evolutionary relationships between the virus, vector, vertebrate host, and ecological niche. So if you look at this tree here on the slide, you have at the far left the 4 serotypes of dengue. Those are hemorrhagic viruses transmitted by Aedes mosquitoes. The JE branch is Japanese encephalitis. Those are primarily transmitted by Culex mosquitoes and involve birds as the reservoir. To the far right, you have the tick-borne encephalitis viruses. And in the middle, with no real close relatives, would be yellow fever virus. Flaviviruses cause different febrile syndromes depending on the virus and the affected patient. Several flaviviruses are neurotropic, for example, West Nile virus and Japanese encephalitis virus, and can spread to the brain and spinal cord, causing severe neurological syndromes, including meningitis, encephalitis, and acute flaccid paralysis. These can result in death or long-term disability in survivors. Other flaviviruses, such as yellow fever, dengue, and Zika, cause visceral disease, resulting in liver failure, hemorrhagic syndromes, and vascular compromise, and can also result in death. Uniquely, Zika virus can infect the tissues of the male and female reproductive tracts, leading to sexual transmission. Zika virus infection during pregnancy can cause injury to the placenta and can transmit to the developing fetus, resulting in placental insufficiency, microcephaly, congenital malformations, and fetal demise. Yellow fever virus originated in Africa and was brought to the Western Hemisphere during the slave trade era, with the first epidemic reported in 1648 in the Yucatan. Over the ensuing 200 years, outbreaks occurred widely in tropical America, the North American coastal cities, and Europe. By the 19th century, it was recognized that yellow fever virus was not communicable person to person, but theories wrongly attributed the disease to atmospheric miasmata. In 1881, Carlos Finley of Cuba significantly advanced the field when he suggested that the mosquito Aedes aegypti was responsible for spreading the disease. Despite multiple attempts, he was not able to prove his theory, but it would serve as the basis for future research. At the end of the 19th century, the United States invaded Cuba during its war with Spain. For every soldier who died in battle, 13 died of yellow fever. The Surgeon General of the U.S. sent Walter Reed and other physicians to Cuba to investigate the cause of yellow fever. Reed's work proved that Aedes aegypti mosquitoes were the primary mode of transmission for the disease, and that yellow fever was caused by a filterable agent found in the blood of infected patients. Currently, yellow fever virus is estimated to cause about 200,000 cases of disease and 30,000 deaths each year, with 90% occurring in Africa. 20 to 50% of infected persons who develop severe disease die. Symptoms take 3 to 6 days to develop and include fever, chills, headache, backache, and muscle aches. About 15% of people who get yellow fever develop serious illness that can lead to bleeding, shock, organ failure, and sometimes death. The disease gets its name from the jaundice that infected patients develop. An effective yellow fever vaccine has been available for more than 80 years. Vaccination is the single most important measure for preventing yellow fever. In high-risk areas where vaccination coverage is low, prompt recognition and control of outbreaks through immunization is critical to prevent epidemics. To prevent outbreaks throughout affected regions, vaccination coverage must reach at least 60 to 80% of a population at risk. The yellow fever vaccine is safe and affordable, providing effective immunity against yellow fever within 1 week for 95% of those vaccinated. A single dose provides protection for at least 30 years and probably for life. Serious adverse events have been reported rarely among those who have been immunized in a few endemic areas and among vaccinated travelers. Scientists are investigating the cause of these. But there are some warnings for the vaccine. For instance, a child should be at least 6 months old. You should not get it if you're pregnant. It's grown up in chicken eggs, so people who are allergic to eggs cannot receive it. It is a live virus vaccine, so people with an immunosuppressed immune condition cannot receive the vaccine. Another issue with the vaccine is because it's produced in chicken eggs, it takes a long time to grow it up. So if we were to have a really large outbreak of yellow fever, it would be hard to produce enough vaccine very quickly to handle it. Dengue virus is spread to people through the bite of an infected Aedes mosquito. These mosquitoes also spread Zika, chikungunya, and other viruses. Dengue is common in more than 100 countries around the world. 40% of the world's population, about 3 billion people, live in areas with a risk of dengue. And in those areas, dengue is often a leading cause of illness. Each year, up to 400 million people get infected with dengue. Approximately 100 million people will get sick from the infection, and 22,000 will die from severe dengue. Dengue is caused by one of any of 4 related viruses, cleverly named dengue virus 1, 2, 3, and 4. For this reason, a person can be infected with the dengue virus as many as 4 times in his or her lifetime. Mild symptoms of dengue can be confused with other illnesses that cause fever, aches, and pains, or a rash. Symptoms of dengue typically last a few days, and most people will recover after about a week. There is no specific medication to treat dengue. About 1 in 20 people who get sick with dengue will develop severe dengue. Severe dengue is a more serious form of disease that can result in shock, internal bleeding, and even death. You are more likely to develop severe dengue if you've had a dengue infection before. Infants and pregnant women are at increased risk for developing severe dengue. A pregnant woman already infected with dengue can pass the virus to her fetus during pregnancy or around the time of birth. Dengue can have harmful effects, including death of the fetus, low birth weight, and premature birth. Dengue outbreaks are occurring in many countries of the world in the Americas, Africa, the Middle East, Asia, and the Pacific Islands. Anyone who lives in or travels to an area with risk of dengue is at risk for infection. Before you travel, find country-specific travel information to help you plan and pack. Again, as I said, 40% of the world's population is at risk of dengue. Dengue has emerged as a worldwide problem since the 1960s. The disease is common in many popular tourist destinations in the Caribbean, including Puerto Rico, Central and South America, Southeast Asia, and the Pacific Islands. In the United States, local cases and limited spread of dengue does appear periodically in some states with hot, humid climates and Aedes mosquitoes, particularly in Florida. Dengue virus disease became a nationally notifiable condition in the U.S. in 2010. Cases are reported to CDC by state, territorial, and local health departments using standard case definitions. Dengue is common in the U.S. territories of Puerto Rico, the U.S. Virgin Islands, and American Samoa. Outbreaks occasionally occur in the continental United States, but nearly all dengue cases reported in the 48 contiguous U.S. states were in travelers infected elsewhere. In 2020 so far, there have been 350 locally acquired cases in Puerto Rico and 64 locally acquired cases in Florida. All the other cases that have been reported in the U.S. were in travelers who were infected elsewhere. Most outbreaks in the United States have been relatively small and limited to small areas, but the types of mosquitoes that spread dengue are common throughout many areas of the United States, so local spread of dengue is possible and does occur. Most people don't develop any symptoms after being infected with Zika virus. And for everyone else, the symptoms are usually mild: a rash, headaches, pain in the joints and bones, and fever. They show up between 3 and 12 days after the initial mosquito bite and resolve within a week. Hospitalizations are uncommon and death is rare. Most importantly, though, scientists have found evidence that Zika causes severe birth defects, most notably microcephaly. The congenital condition is associated with a small head and incomplete brain development, part of a pattern of Zika-linked birth defects, including eye damage and muscle problems. While the virus is passed mainly through mosquitoes, people can catch it through sexual intercourse, which is why there's a Zika concern for men, too. Male partners of pregnant women and women who want to become pregnant are advised to use condoms or avoid sex for the duration of a pregnancy or 3 months before conceiving if they've traveled to areas with current or past Zika. The urgency to fight Zika reached a fever pitch a few summers ago when a massive outbreak struck South and Central America and the Caribbean, causing more than 500,000 suspected cases and more than 3,700 congenital birth defects. Some scientists suspect a mutation in the virus made the disease more harmful and triggered the epidemic. It also landed in an ideal climate where tropical mosquitoes, namely Aedes aegypti and Aedes albopictus, helped propel Zika to such large numbers. But then, almost as rapidly as it appeared, the virus started to decline sharply in its hotspots and all but disappeared in the U.S. In 2016, Puerto Rico, the U.S. Virgin Islands, and America Samoa saw more than 36,000 cases of locally transmitted Zika virus. By 2017, the number had dropped to 665. In 2017, the continental U.S. saw only 7 cases of local mosquito-borne Zika, down from 224 cases the previous year. Most experts say the sharp decline in Zika cases is due, at least in part, to herd immunity. When enough people become immune to a virus, whether through vaccination or natural immunity, then that disease can't easily travel from person to person. But over time, the benefits of herd immunity wane as more children are born, providing fresh tinder for the next epidemiological wildfire. Researchers are also unsure whether those infected with Zika are immune for life or just for a period of time. Most experts predict future outbreaks because the underlying conditions that enabled the epidemic, crowded urban neighborhoods whose residents are too poor to afford insect repellent or window screens, remain a problem in much of the developing world. Also, the mosquito has developed a particular fondness for human blood and has adapted really well to urban living. So we shouldn't become complacent. Zika has probably not disappeared forever. Zika hasn't disappeared entirely. Last year there was an outbreak in northwestern India. There are concerns about further spread, but spotty disease surveillance systems are likely to overlook smaller outbreaks. And that means that just because Zika isn't popping up on epidemiological maps in a country doesn't mean it's not there. Let's move on to the genus Hepatovirus. Hepatitis C virus, the only member of this genus, was identified in 1986. Interestingly, it was revealed by the development of diagnostic tests for hepatitis A virus and hepatitis B virus. So for a number of years before hepatitis C was isolated and identified, there was a recognition that there was a third viral agent causing hepatitis, but they didn't know what it was, and actually for a number of years they just referred to it as non-A, non-B hepatitis. The infection can be extremely serious, causing persistent infections with a majority of cases progressing to chronic hepatitis. People with chronic hepatitis can develop cirrhosis of the liver and also liver cancer, hepatocellular carcinoma, or HCC. Most persons who acquire hepatitis C virus will develop chronic infection. Following acute infection, hepatitis C is very successful in establishing persistent infection by evading the immune system. Although the mechanism for the high rate of viral persistence is not completely understood, several viral and host factors likely play a significant role. The rate of viral production is high, and there's a lack of proofreading by the viral polymerase, which leads to enormous genetic diversity, which in turn creates major challenge for the host immune response. This broad genetic diversity contributes to the likelihood of developing chronic infection. But host factors are also involved in the ability to spontaneously clear the virus. For instance, people who are younger are more likely to clear the virus. Men have a higher risk of developing chronic infection compared to women. African Americans have a higher risk particularly African American men, of developing chronic infections. They're less able to spontaneously clear the virus. Um, Other kinds of conditions also play a role. Having a normal immune system, of course, is a factor in your favor. But there's an interesting study that showed a particular genotype associated with the clearance of the virus. Individuals with the CC allele of the IL-28B genotype are more likely to spontaneously clear hepatitis C than those with the CT or TT genotype. In one report involving over 1,000 individuals, those with the CC genotype cleared the virus 53% of the time, compared with a clearance rate of only 23% of those with the TT genotype. So this is a nice example of how your genotype can affect your ability to clear this virus. The natural history of chronic hepatitis C infection has not been fully delineated, primarily because most persons are not identified at the time they have acute hepatitis C infection, and most remain relatively asymptomatic for at least 15 years after becoming infected. Further, it's been difficult to design studies that convincingly define the natural history of chronic hepatitis C infection for multiple reasons, including the difficulty of accurately establishing the time of initial acquisition of infection, which sets the timeline for determining duration of infection, and the necessity to follow patients for decades to see clinical sequelae. Because remember, cirrhosis and liver cancer and so on, those kinds of outcomes develop years after the initial infection. There are a number of factors that promote the progression or severity of hepatitis C infections. We've already talked how age and gender can impact the ability to clear the virus from the body. Co-infection with HIV tends to result in a poor prognosis for a patient, as does co-infection with hepatitis B virus. People who have insulin resistance and or diabetes mellitus, that is diabetes 2, are more likely to progress to severe infection. Use of marijuana, use of alcohol, and use of coffee are also not recommended for anyone who has a hepatitis C infection, because it can lead to progression of disease. Hepatitis C is the number one cause of liver cancer and liver transplants in the U.S. So it's brought on by a virus you can catch if you come into contact with contaminated blood. You can also get it from an unclean tattoo needle, for example. Sometimes it spreads during sex. For decades, the only kind of treatment that people could get was a series of painful shots of interferon and taking a pill called ribavirin. These drugs didn't target the virus that made you sick. Instead, they amped up the immune system so you'd fight it when you do get the infection. But the treatment didn't always get the virus out of the body. Cure rates hovered around 50%. And people who stuck with the year-long treatment, and not all did, had to live with the chemo-like side effects of the interferon treatment. These days, more and more people can get rid of the virus by simply taking a pill at home for just a few weeks. There are several ways to do it without having to get shots. Now, there's no one-size-fits-all option. There are many different types or genotypes of hepatitis C virus. Type 1 is the most common. This is important to understand if you were going to talk to your doctor Not all meds work on all types. Which medicine is best for a patient depends on how much liver scarring or cirrhosis they have and the genotype of the infecting virus. There are a number of ways that individuals can become infected with hepatitis C. Injecting drug use is a major risk factor. Transfusion or transplant from an infected donor can also result in transmission of the virus. Occupational exposure to blood, primarily through needle sticks, can be a cause of this. Iatrogenic causes such as unsafe injections can cause this. We talked about these kinds of exposures earlier in the semester. Also, a child who is born to a hepatitis C infected mother can become infected, primarily through exposure to the mother's blood at the time of birth. And sex with an infected partner, particularly for individuals who have multiple sex partners, would be at risk. And so in the pie chart you can see injecting drug use is responsible for about 60% of the exposures in the U.S., with sexual exposure amounting to 15%, and transfusion can also result in a significant number of cases. But we do have a good screening program now, so that mode of transmission risk has gone down substantially. In this graph, you can see how screening has greatly reduced the risk of acquiring hepatitis C through transfusions. And across the bottom of the graph, we just have year, and we're looking at the percent of recipients infected. And initially in around 1970, they developed a screen for hepatitis B surface antigen. So even though hepatitis B is a different virus, screening for that virus helped screen out people who might also be positive for hepatitis C, because the two viruses share the same modes of transmission and the same risk factors. And over time, the rates remained constant until they started screening for HIV risk factors. In the 80s, when HIV was recognized and they realized how it was transmitted, they started screening people even before they had an actual molecular test for HIV. They did donor screening through questionnaires, and the risk factors for HIV and hepatitis C are very similar. So by screening out people with HIV risk factors, you were also screening out people with hepatitis C risk factors. Then we came up with a actual test for HIV, and then better tests for hepatitis B, and then in the 90s they actually developed a test for hepatitis C virus. And now, with a combination of donor screening through questionnaires and blood screening for Hep B, HIV, and hepatitis C, we've greatly reduced the risk of post-transfusion hepatitis C infection. Hepatitis C is a risk factor for people who are working in various kinds of health fields, people who might have an occupational exposure to blood. So there's post-exposure management steps that can be taken. So if an individual has a known exposure to a patient's blood, for instance, like with a needle stick, the healthcare worker should be tested within 48 hours of exposure for hepatitis C antibodies. And if they're positive, you want to follow up with a test. And if they continue to be positive, then you refer them to pre-existing chronic infection care, because those individuals already had hepatitis C infection. If they're negative, then you want to do follow-up testing. You want to follow these people for about 3 weeks after exposure. If they turn up positive after 3 weeks, then you refer to care. Remember, there are very effective treatments for hepatitis C virus, and the sooner treatment is started, the better. The less likely it is to have progressed to chronic infection, and less likely it is to have caused any liver disease. And if the individual after 3 weeks from the exposure is negative, then you can stop worrying. There's no risk. Okay, now let's finish up by looking at the genus Pestivirus. Pestiviruses infect pigs and ruminants, including cattle, sheep, and goats. Moreover, pestivirus sequences have been detected in samples from bats and rats by next-generation sequencing, but infectious pestiviruses have not yet been isolated from these host species. There are no invertebrate hosts. Transmission occurs by direct and indirect contact, such as through nasal or urine secretion, feces, and contaminated food, and transplacentally. Infections may be subclinical or produce a range of clinical conditions, including acute diarrhea, acute hemorrhagic syndrome, acute fatal disease, and a wasting disease. Transplacental infection can result in fetal death, congenital abnormalities, or lifelong persistent infection. Fatal mucosal disease can occur in cattle persistently infected with non-CP viruses when a cytopathic virus is generated by mutation or introduced by superinfection. Pestivirus infections of livestock are economically important worldwide.